Hello, everyone. Hi. Welcome to today's webinar. So we're just waiting for people to enter. Hello. Okay, so we'll just wait for a few seconds and then we'll begin. All right. Okay, so hello again, everyone. Thank you so much for joining us from everywhere around the world. I see that we have attendees from Asia, from Europe, and also from the US. Thank you so much for joining us today for another MaxWell webinar. I'm so happy to see you again, and I hope you will enjoy today's webinar. So for today's talk, we have the following speakers. On our side at MaxWell Biosystems, we have Szilárd Sajgó, our R&D scientist, and he will be introducing MaxWell Biosystems and also our technology. Our guest speaker for today is Alessio Buccino, currently a postdoctoral fellow at ETH Zürich. So Alessio is currently at the Bioengineering Laboratory at ETH Zürich, BSSE department. After receiving his PhD at the University of Oslo, he then proceeded his work at ETH Zürich. So Alessio is currently combining neurotechnology, computational neuroscience, neural engineering to improve electrophysiological techniques in neuroscience. And he's particularly interested in advanced recording and stimulation modalities to probe neural activity in biophysically detailed computational models of neurons and large-scale analysis of electrophysiology recordings, especially spike sorting. So for today, you will hear about Alessio's fantastic work on the SpikeInterface, which is a unified framework for spike sorting. So this session today is a follow-up of our first spike sorting webinar, which you can also find from our webpage. We will also share that to you later on. So for today's webinar, we will end with a Q&A session together with Alessio. So feel free to ask your questions here in the chat that you have on the right side. And we would also be sharing some information on what's next for MaxWell Biosystems at the end of the session. All right, so let me welcome Szilárd Sajgó. Hello, Szilárd.
Hello, Mary.
Hi, so the floor is yours.
Hello, everyone, and welcome to our MaxWell webinar. And let's just start at what do we do here at MaxWell. So at MaxWell, our mission is to provide customers electrophysiology systems and solutions to advance their scientific discovery and to accelerate the development of new drugs for neurological disorders. Our core expertise lies at the heart of our core expertise, like microelectrode array platforms, hardware and software solutions, and we have applications using MEAs represented in more than 17 journal papers so far. Our customers include academic customers, contract research organizations, and of course the pharmaceutical and biotech companies around the world. What is our technology about? So classically, you might know of two distinct ways of characterizing cells, right? So the electrophysiological properties of cells, let's say. So one would be the gold standard, which is patch clamp electrophysiology using a glass micropipette. And the other, for instance, could be imaging that allows you to see different cellular morphology, cellular subcompartments, and so on. So our technology basically falls between these two techniques. And here you can see in the middle an example of one cell recorded using our high-density MEAs, and you can see not only the activity of the neuron, but also its morphology. Now, what do we actually measure with this technology? Well, at heart, this is an electrophysiology tool. So compared to patch clamp that records intracellular signals, our sensors are located outside of the cells, but they are recording extracellular action potentials. And with the application of ultra-high density MEAs, we have multiple of these tiny electrodes at super high density. Thus, we can detect signals from one single neurons in multiple points. Let's see, along its axonal shaft, its axonal initial segments, its dendrites, and so on. Now, our system has several key advantages when over classical MEAs that have been used for the past 40 years. In conventional MEA systems, where you can have cells between electrodes that are not detected because of the high electrode pitch, or you can have multiple cells on one electrode, thus you can only measure average activity. In the MaxWell MEA system, you will always have an electrode in the correct position, and thus you can record theoretically every single cell in your culture. And of course, because these electrodes are super small, they're only 10 micrometers across, and they're really close to the signal source, you can record very small amplitude signals, like, for instance, axonal signals. Our products include two systems. We have the Max1, which is a single well format, and then we have the Max2 mainframe, which is basically an incubator that can not only record your data, your electrical data, but can also maintain the physiological conditions needed for extended recording periods, like temperature, CO2, and so on. And at the heart lies the MaxWell Biosystems HD-MEA chip, which is the same for both of the consumables. And it contains 26,400 platinum black coated microelectrodes. It has an 8mm square recording area. And as I mentioned before, it has really low noise readouts. And we also need to mention that these electrodes are not just capable of recording electrical signals, but they're also capable of stimulating your cells to provide electrical stimulation to your samples. Now, of course, centered to the system is MaxLab Live. It's the software that we use to visualize and analyze the data. And in the first instance, once you start your experiment, you can have different visualization panels. You can have the electrode views. You can see where your cells are located in the electrode in physical space. You can have an electrode raster view where you can see whether there's any synchronous activity in your culture. And of course, you can also have a color map heat view. For instance, this is an eggplant retina presented with different light stimulations. As you can see, when the light stimulation appears in the field of view, then you have action potentials happening. Now, of course, the software also contains several easy-to-use assays. And these are the activity scan, the network analysis, and the exon tracing. And we detail them a bit. With the activity scans, what you can achieve is you can answer whether your neurons are active. You can create a whole sample activity map, basically label-free, where you can analyze the sample's firing rate and amplitude. And as you can see here on the left, you can see the microscope image of the neurons. And on the right, you can see a functional image of your neurons. So basically, then you can be able to compare these two. Now, once you performed an activity scan, you can analyze and perform a network connectivity assay, where you can answer the questions whether neurons are interconnected. You can track neuronal bursting features, and you can analyze the synchrony of your bursts. Now, if you really want to analyze, let's say, the activity of your neurons at single cell resolution, of course, then we can perform an axon tracing assay. And this allows you to analyze the health of your axons, whether you can see whether your axons are functional. You can also visualize the axon. You can see where the arborizations are. And you can also automatically analyze the propagation speeds of the axon potentials along the axons. And of course, this can be very beneficial if you have, let's say, a diseased phenotype axon. So these are the three assays that can be applied for neurons. But of course, the system is also suitable for many other applications, like recording from muscle cells, from cardiomyocytes, brain organoids, acute retina and brain preparation, neuronal spheroids, and so on. Now, When you have all this neural data that you acquire, and for instance, if you are analyzing more complex tissues with the MEA, like a retina expat sample, unlike in cultures where you might have 2 or 3 cell types, in an acute recording like the retina or a brain slice, you can have anything from 1 to 40 different cell types. And here the notion of cell type is not just cells with different morphologies and different connections, but also different physiologies. So now how can you turn, for instance, the electrode raster that you have seen before into a cell raster where you can clearly say that the activity coming from these electrodes is assigned to one particular cell. So basically what you need to do is you need to perform a process called spike sorting, where you are going to go from the channel raster to actually the neuronal raster. Because as you can see here on the left, if you just look at the channel raster, then you can look at, for instance, different light stimulations in different directions. You will not be able to say and to distinguish which cell has what kind of response properties, temporal response properties. Now, if you perform the spike sorting process and you assign particularly the spikes to the different neurons, then you'll be able to say that you have different neurons in your culture or your retina that you have recorded with different temporal properties. So this is the key advantage of performing spike sorting. Now, what lies at the heart of this process, spike sorting? You might hear it or read about a lot in different papers that they just use spike sorting and these are the results. Now, to explain it in a very easy notion is just imagine you have a room full of people. There is a large chaos. Everybody is talking. Now, in the biological sense, you can say that the people are retinal ganglion cells, and then the microphones are electrodes. And now, you as the spy or the experimenter enters the room, and you are given the task to reconstruct the individual discourses. This basically being a classical cocktail party problem. So your task as a spy is to identify how many people are talking, how many neurons you have. For this, you would need to sort the voices based on their shape, their frequency profile, and so on. But you will encounter several problems. If you have only a few microphones, of course, you will have people that speak louder, some speak softer. People to the microphone that are closer to the microphone will be heard more clearly than the ones far. So more noise will also be recorded on each microphone. And one way to improve on this is basically using a high density MEA, meaning you use more microphones, and thus you can clearly and easily reconstruct the signals coming from individual neurons. Now, of course, having more electrodes creates comes with several computational challenges. And as Alessio will mention a bit later, the main challenge is that there's no one spike sorter that works for all. There's a huge array of them, and each one has their tiny bits of advantages and a tiny bits of disadvantages. And you, as the end user, might not know precisely on how to optimally perform the sorting, how to set your settings, and so on. So this is where Alessio's very beautiful work comes into play, where he has developed a unifying platform where you can perform spike sorting with different algorithms and compare the results. Thus, this would give you more certainty that your results are correct and indeed coming from single cells. And with this, I would like to welcome Alessio, and I will just hand the microphone to him. Welcome, Alessio.
Hello. Hi, Szilárd. Hello, everyone. Let me share my screen quickly. All right. Can you see my screen? I guess. I wait another 5 seconds so that we're sure that the sharing is working. All right. So hello, everyone, and thank you for attending this webinar. And I would like to thank MaxWell for organizing this webinar and inviting me as a speaker. Today, I will be introducing and explaining what SpikeInterface is about. So SpikeInterface is the result of a very productive and wide collaboration between myself at the ETH Zürich and previously at the University of Oslo, the University of Edinburgh, the Center of Research and Neuroscience in Lyon, the Flatiron Institute in New York, and the Allen Institute of Brain Science. So Szilárd already greatly introduced what these extracellular recordings look like, especially focusing on these high-density electrode arrays that MaxWell produces. But I just want to mention that there are many different kinds of extracellular recordings that you could get. So the simplest version would be using microwires or tetrads. So these are simply usually tungsten electrodes that are bundled together and inserted in the brain. And so usually they're called tetrads because 4 wires are bundled together, and that improves the separability of different neurons, and the recordings would look like this. And these kind of probes were developed around the 80s. And then silicon probes came. So silicon probes are metal electrodes which are embedded in a silicon substrate, and they are purely passive. So then the metal electrode will have a wire that then goes out of the head stage and is connected to an acquisition system. And in recent years, I would say in the last 10 years, we had the development of these high-density microelectrode arrays, both for in vitro application, like the MAX1 and the MAX2 system, and also for in vivo application, for example, like the NeuroPixels probe. And the main evolution of this probe lies in the fact that the electronics sits together with the electrodes. So these kind of probes can achieve a much higher spatial density and also a much lower noise level, while the main bottleneck for the other kinds of probe is in the connection. So each electrode in this case, in case of silicon probes and in case of tetrads, must have like a specific wire that will need to be connected to the acquisition system. But in this case of high-density microelectrode arrays, the electronics sits with the electrodes, and so the signal from different electrodes can be sampled, and then what comes out of these head stages is a digital signal, which require a lower amount of wires, so they can achieve higher density. And if you're familiar, if in your career you have encountered this kind of electrodes, then you're going to be familiar with spike sorting. So if you want to extract some meaningful information about what single neurons do in your recording, you have to do some spike sorting. And spike sorting, so it's an essential step in extracellular electrophysiology analysis, and it allows us to extract from the mixture of signals coming from many neurons and recorded from your extracellular electrodes. Spike sorting allows you to infer the spike train of isolated neurons or units. And this is a very complicated problem because there are many aspects of neural activity which makes it quite hard for automatic algorithms to do this processing. And this is why in the last 10, 15 years, there's been a huge development of spike sorting tools that I'd like to call a spike sorting jungle. You have Cluster that was developed in 2016, Spike and Circus, the Kilosort family, which is quickly evolving, Ironclast, the HD sort, herding spikes, and so on. There's really a plethora of tools so that people ran out of names, and so they're calling their spike sorted yet another spike sorter. And so with this large variety of tools that you could try out for your own data, it's okay that sometimes you might feel like this, right? A little bit lost, and rightly so, because this multitude of tools actually is a problem for the neuroscience community. And let's see why. There are some unaddressed challenges that comes out from this large availability of different tools. And the first one is that mainly they are incompatible with different file formats. So some spike sorters are compatible with some acquisition systems, other spike sorters are compatible with other acquisition systems. And so labs tend to focus on the spike sorting tool that define that they're compatible with the acquisition system that they use, and it is difficult to build a software that tries to accommodate all the different file formats out there. The second complication and issue is that it's very hard to run multiple spike sorting algorithms for several reasons. So the first reason is that they're usually written in different languages. Some are written in Python, some are MATLAB based, there are some C and C++ code out there for spike sorting. And also each spike sorter comes with its own configuration files, with its set of parameters. So it's really a tedious task to set up a spike sorter. And that usually bring about the fact that a lab that runs experiment with extracellular recordings, usually it's capable to set up one pipeline with one spike sorter, and that takes time and effort, and then they stick with it forever, right? So it's kind of hard to switch because the barrier in order to set up another spike sorter is quite high. And another side effect of these different spike sorting tools and different compatibility issues is that there is not a rigorous benchmarking of current algorithms. If you look at these, the papers presenting the different spike sorters, you see that spike sorter 1 compares with spike sorter 2 and 3, spike sorter 3 with spike sorter 4 and 6, spike sorter 2 with 3 and 2. So it's a little bit complicated to make a sense out of this jungle. And the third problem is that since setting up a spike sorter requires, as we'll see in the next few slides, setting up some custom pipelines, this kind of harms the provenance and reproducibility of science. So there would be a lot of this process of setting up spike sorter on a lab-specific experiment. It's prone to errors. And 10 years from now, who knows if we will be able to reproduce the data presented today. So in order to address these challenges, we produced a Python package that's called SpikeInterface. And SpikeInterface, the main goal of SpikeInterface is to create a common ecosystem that can encapsulate all the spike sorting software which is available out there. And with the SpikeInterface, you can very easily run multiple spike sorters on the same data set. And then we were able to bundle functionalities for the entire electrophysiological pipeline. So from pre-processing to post-processing, we have validation and curation tools to improve your spike sorting output. We have tools to provide spike sorting comparison, and I will show later how these work in practice. We have data visualization widgets, so you can, along your analysis, you can visualize your raw data, your filter data, or pre-processed data, the spike sorting output, and so on. And we try to do this in the most reproducible way possible. So every step in a SpikeInterface is logged, and it could be reproduced on a different machine. So let me try to explain what was the main concept. So these, the three challenges that I introduced before are mainly software engineering challenges. And so the manual workflow usually starts from the raw data file, which comes from an acquisition system. And then, as I said before, it requires some custom codes to interface the raw data to the requirements that a certain spike sorter have. So the requirements can be both in terms of the file format that needs to be injected to the spike sorter, or other kind of metadata that are needed for the spike sorter to run. For example, how many channels are there in the data, what's the location of your electrodes, what's the sampling frequency, and so on. And usually these information are very spike sorter specific. And then the spike sorters spit out their sorted data in their own sorter-specific format, right? What we try to do in SpikeInterface, and I think we managed to do it in a certain way, was to provide a layer of abstraction between the data formats, both the raw data and the sorted data, and our API, or application programming interface. So basically, the idea is that we have some Python objects that are called extractors that interface directly to different file formats, but they provide a common interface. So this recording extractor, for example, will interface with a file format, and from this object, you would be able to extract in a standardized way, and that's basically the key concept, all the information that are relevant to spike sorting. For example, how many channels are in your recordings, what the sampling frequency, what are the channel IDs, and of course, we need to be able to extract the data from the file. Once we have the standardized interface, we can interface to any spike sorter also in a standardized way. And the spike sorter will spit out its sorted data, but we have another object in SpikeInterface called sorting extractor, which will also interface to this sorted data in a standardized way. And so with this sorting extractor object, you will be able to retrieve the information that are relevant for further downstream analysis, such as how many units are found by the spike sorter, what are the unit IDs, what are the spike trains, and so on. And this standardized interface allow you to do something like this. Instead of running a single spike sorter on your data with your custom code, since we have this standardized interface, we can run, for example, 3 different spike sorters and check out what's the output, and then pick the one that we think it's more accurate and more relevant for our purposes. And we can also keep leveraging this standardized interface to add pre-processing and post-processing steps to our analysis. For example, before running the spike sorter, you might want to manipulate somehow your input recordings in order to improve the spike sorting performance. And the same from the spike sorting output, you might want to run some post-processing steps in order to clean up the output of spike sorting. And then you can store or export your data in a few different formats. So this is a bit an abstract notion of SpikeInterface pipeline. But before showing a concrete example, I just want to mention that currently we can interface with many different technologies. So SpikeInterface can read data from each of these acquisition system and file format. We have over 10 spike sorters that are implemented, and this can be run with a single line of Python code, as I will show in a few slides. And we can interface also to spike sorted data, so the ones that come out from the spike sorting algorithms, also other spike sorting data that have been commonly used in the neuroscience field. So let's take a look at a concrete example of a SpikeInterface pipeline. So what could we do with SpikeInterface? Let's assume we have our original recording. It could be an OpenIFIS recording, which is this open source acquisition system. It could be a recording from the MaxWell system, any file format which is compatible with SpikeInterface. The first step would be to load the recording into SpikeInterface, and then we might want to provide some probe information to basically make spike sorting more accurate. For example, in case we have, let's say, 16 channels with 4 tetrads, we want to tell the spike sorter that each channel group, so each tetrad should be spike sorted separately. And with this probe file, we can also provide the geometry of the different electrodes. And this is very important in case you have these high density probes, because they definitely improve the outcome of spike sorting. And then we might want to do some pre-processing. So this could be 2 common pre-processing steps that I showed before. We can have different kinds of filters, like a bandpass filter. We also implement notch filters and whitening and so on. We can also try to remove some of the common noise in the recordings by applying some common median re-referencing of the data. And then when we are happy with our pre-processing, we can finally run spike sorting. In this pipeline, we would run Mountain Sort 4, which is one of the available ones. And once we have our spike sorting output, we would maybe curate the output saying, I want to remove all the units with less than 100 spikes because I know I have a very long recording, so I'm not interested in those. And finally, we can export both the recordings and the sorting output to an open source software called Phi, which provides a very nice visualization and the capability to perform manual curation on your data. So, for example, to merge or split units, to remove noisy units, and so on. So this pipeline is made of 1, 2, 3, 4, 5, 6, 7 steps. And in SpikeInterface, it would look like this. It literally translates to 7 lines of Python code. And the nice thing about this API is that it's pretty self-declarative, right? So it's really straightforward to understand what each line of code is doing. First, we start by loading our recording, OpenIFIS recording extractor. Then we load our probe file. We perform some pre-processing with a bandpass filter and then a common reference using the median as reference. And then we can run mountain sort with 1 line of code. And after we are done, we can remove, so we can threshold or curate the spike sorting output based on the number of spikes, and we can choose our spike threshold. And finally, we can export to phi, and in this case, we have to combine the recording, the sorting that we have already curated, and we just have to provide an output folder. Okay, so this makes it quite easy to construct these processing pipelines. But another thing that is quite interesting now is that we have a unified framework that allows us to run several spike sorters on the same data. And this opens up for a whole set of interesting analyses that go into the meta-analysis of spike sorting output direction and towards the benchmarking of spike sorting tools. And in the paper that, okay, but paper is the next slide, but let me say a couple of words on how we can do this spike sorting comparison. So first of all, in order to assess the quality of some spike sorting output, you would need some ground truth data. And with ground truth, I mean that you need to know exactly when there was a spike for a certain neuron. And this is quite complicated, but there are a few ways to get this kind of data. And one way is experimental. So for example, we could have some extracellular recordings, and then we could patch one neuron outside or on top of these extracellular probes. So the patch signal will give us the exact spiking times for that neuron. And then we can look at the spike sorting output and check which sorted unit basically matches with the patched unit and assess the performance of the spike sorting of that specific unit. So in this case, we would be able to benchmark 1 unit at a time. But another possibility is to use simulated data. So we have very good models to simulate neuronal activity. And so in that case, we can simulate complicated recordings with hundreds of neurons. And in this case, we can really benchmark the performance of all the spike sorting output. And we would do that by first computing an agreement score between the ground truth spike trains, each of the ground truth spike train, and each of the sorted spike train. And then we would find the best match with this agreement score. And once we have the best match, we can compare the matched spike trains and extract different performance metrics such as precision, recall, and accuracy. And we can also classify the spike sorted units as being well detected. So they actually are found both in the ground truth spike in the ground truth spike trains and in the sorted one. We could tag them as false positives if they are wrongly found by the spike sorter. They can be redundant or over-merged units, and these are a bit more complicated concepts that I won't go into now. And we can also start comparing multiple spike sorters at once, even without ground truth. So we could, for example, take some experimental data, run several spike sorters, and then compare the output of different spike sorters. So in this case, we perform a pairwise comparisons between the output of all spike sorting pairs, and we compute a graph in which each node is a unit found by a spike sorter, and each edge is the agreement score between the different units of the different spike sorters. And then we can interrogate this graph, asking how many units are in agreement with 1, 2, 3 spike sorters, and so on. And we did that in the paper that came out in eLife in November. So we started off with neuropixels recording from the Allen Institute of Brain Science. It was a 15 minutes recording with 246 active channels, and the results were quite impressive. So we ran 6 popular spike sorters: Herding Spikes, KiloSort2, IronClust, 3D Clue, Spike and Circus, and HD Sort. And in total, all the spike sorters, we pulled together the units found by all the spike sorters. We had more than 1,400 units. And out of these 1,400 units, only 33 units were in agreement with all spike sorters. Bam. So there's very low agreement between spike sorters on real data. So be aware of this. But then we were not so negative about it. We tried to basically have some optimistic view. So we tried to use the combination of several spike sorters to try to see if using more than one spike sorter could actually improve the spike sorting quality. And in order to assess the performance of this approach, we moved to simulated data. So we simulated a NeuroPixel dataset using the MEAREC simulator that you can check out. You can just Google it, you'll find it. It's a 10 minutes recording. And in this case, we know we have 250 ground truth units because these are simulated data. And since we know the ground truth spiking activity, we can now compute all this accuracy, precision, and recall. So we can see that here KiloSort2 seems to be very, very good in detecting most of the ground truth units. But on the other hand, we can also, as I said before, classify the units as being well detected, false positives, or redundant. And we can see that KiloSort does a very good job as detecting the good units, but it also finds a lot of false positives. So this means that it would take some time if you are an experimenter to manually curate the data in order to proceed with your analysis. And we can see different kinds of behavior across the different spike sorters that we tested. And we also did the same analysis that we applied on the real data. So we compared the output of all the different spike sorters, and you can see the results here. In this case, 139 units were in agreement with all spike sorters, so better than the real data. But I also have to mention that this simulated data was relatively simple with respect to real data. We didn't model some complicated aspects of experimental data like drifts or high spike synchrony in the spike trains and so on. But one thing that we found, and we can see it here in the bottom graph, is that most of the units that are false positive units, so the ones that are not matched to any ground truth units, are only found by one spike sorter. So here on the x-axis is how many spike sorters agreed on the units, and we can see that the false positive bar, the red one, localizes almost all the time with the one. So it's only found by one spike sorter. So we thought that this could provide a good idea to combine the output of different spike sorters in what we refer to as ensemble spike sorting. And in order to prove that this could be a viable solution, we turned back to our initial experimental data, and we asked two experts to manually curate the spike sorting output. And we can see how the curation looked here. So we can see that many units, we started from the KiloSort2 output, so that's quite important. And then the two curators found 207 plus 19 units that both basically classified as good units. And there were already some disagreements between the two curators, which is very common. So spike sorting curation, it's quite a subjective step. So that's why also we are trying to push into fully automated spike sorting for reproducibility. But if we look at the raw spike sorting output of the different sorters, we can see that basically only for KiloSort, for example, only 35% of the units found by the KiloSort2 are in agreement with the units that came out of the curation. You can see here the two colors are curator 1 and curator 2. So over 50% of the units that KiloSort2 spit out, and also the other sorters, are doing actually even worse. Of course, KiloSort2 here is a little bit facilitated because curation started from the output of KiloSort2, but still less than 50% of the units would be actually kept for further analysis. When we actually use the units that are in consensus with at least 2 spike sorters, we see that these, the agreement with the manually curated data goes up above 80% for all cases, right? So we suggested that using multiple spike sorters could actually be a way to automatize a bit more the manual curation step. And because there is a very large agreement between the consensus-based unit or the output of this ensemble spike sorting and the output of hand curation on spike sorted data. I also want to mention that we have other features in SpikeInterface that I didn't present here. And one actually very important one is that we provide functions to compute a wide range of quality metrics. So far, especially in this slide, right, I presented a quantitative assessment of spike sorting performance, but because we knew the ground truth activity, right? But most of the times, actually all the times when you analyze your data, you don't know how many neurons you have in your recordings and when they're firing an action potential. So you can't rely on that kind of comparison. But you can actually compute a whole set of quality metrics that could give you some indications on how good is the spike sorting output on a unit base. So this could provide guidance to further accept or reject some units that are the output of your spike sorting. Then we have several visualization widgets for spike trains, templates, PCA projections, and many more. And then SpikeInterface also makes it very easy to manipulate your recordings. For example, if your experimental setup, if in your experiment you have 3 different recordings on the same animal but on different conditions, or on the same cultures but you apply some drug and so on. So you have 3 different files, but you would like to spike sort them together. So you can concatenate the recordings in time. You can also cut them to, for example, synchronize it with other signals and so on. And finally, I want to mention a twin project of SpikeInterface, which is called the Spike Forest, and it was also published in eLife in last year. And Spike Forest is basically, it's based on the SpikeInterface engine, but Spike Forest tried to benchmark all available spike sorters on a wide range of ground truth data. So in the Spike Forest website that you can find here, and I can also show it later, you'll see different kinds of studies. It's over 1.3 terabytes of data. They're either paired recordings or synthetic data, so simulated data, some manually curated ones, and some hybrid recordings. And then we ran systematically 11 different spike sorters, and we can explore the output as a single unit at single unit resolution. And it's interactive, so we can change some thresholds. We can display the accuracy, precision, and recall for all the different spike sorters. And we think that these kind of tools over time will provide also some guidance to the users in order to choose which spike sorters might work better in their case, and also which it's also pushing the spike sorting field to improve their tools in order to have better results on these kind of platforms. And so with this, I would like to thank you all for the attention, and especially I would like to thank my collaborators, Cole Hurwitz, Samuel Garcia, Jeremy McGland, Josh Siegel, and Matthias Senning for basically being having worked with me over the last 3 years on this tool that we developed. And then I would like to thank you again for the attention, and have fun spike sorting.
Amazing, amazing. Thank you so much, Alessio. This is really fantastic. With the Spike interface, you really allow any or all spike sorters to be accessible to everyone using technologies recording electrophysiological data. So this is really cool. Thank you so much. All right. So now we are going to our question and answer portion. We do have a lot of questions already here. So maybe I can just start. All right. So Okay, so here within the presentation, actually Szilárd showed this presentation wherein there are multiple neuronal types from the retina that you can identify. So there is literature on action potential characteristics, kind of characterizing, the different types of neurons, probably the shape of the signal or the firing rate of the signal, and so on. Does the spike sorting parse waveforms into probable neuronal types, is the question.
Not by default. So by default, spike sorting is actually, sorry, I have to go back here. So basically what spike sorting does is just this, right? So spike sorting takes some raw data and it spits out a set of spike trains, right? What you can do also in SpikeInterface is to basically merge the raw data and the spike times to compute waveforms that are related to the different units. From the waveforms, we can compute some extracellular action potential that are called also templates or footprints. And then also in SpikeInterface, we can extract some common features in these templates, like, for example, what's the peak to trough duration or the full width out maximum, the repolarization or recovery slopes, the peak to trough ratio. And these kind of features can be very useful to further characterize the cells that we record from. It's relatively straightforward, especially in vivo, to distinguish between excitatory and inhibitory cells, because usually excitatory cells have a broad waveform, while inhibitory cells are also called fast spiking narrow, and they have these narrow waveforms. And I guess this approach could be also used for specific use cases, like to try to characterize different retinal types, neuron types in the retina or in cerebellum. Of course, each brain area will have its own wide range of neuronal types. So there's not like one single tool that will allow you to do that, but at least you can compute these kind of features in a SpikeInterface and then proceed with the analysis.
All right. Okay. So so you are getting really a lot of questions from everywhere around the world. Thank you so much for staying here. Next one is a question about MaxWell datasets. So this is I'm also curious to hear from you, because I know you have experience with our data. Which of the spike sorting algorithms currently supported in SpikeInterface would be best for processing data from our HD-MEAs?
Good question. So, no, I would say, so this, the MaxWell data, the MaxWell MEAs are relatively similar to these NeuroPixels data. So they are high density and the spacing between electrodes is similar. And in this, in the paper, we only chose the 6 spike sorters that actually support this kind of data. So they need to be able to have at least 1,000 electrodes, and they should be parallelizable and fast. I mean, I usually use both Kilosort 2, now Kilosort 3 came out, but as you've seen here, then it requires some curation. Also, Ironclast could work well. HD-Sort was developed specifically for these probes, but it also might output some units that are not the real units. SpikeInCircus is the same. Maybe you could try. I haven't tried it on MaxWell data. And by the way, you can import the MaxWell data as we imported the OpenIFIS data. Is a one-liner, Max 1 or Max 2. And then you can just give it a try and see what works best. It also depends on the configuration that you use, right? If you have a dense configuration or a network activity, that might really affect the performance and also the choice of your spike sorter.
And maybe also the type of neuron or sample that you're recording from.
Exactly, yeah.
All right, thanks a lot. Next, there are lots of compatible data with SpikeInterface. Are Mobius and pClamp10 datasets compatible? I think these are intracellular, yeah?
No, so SpikeInterface, it's only for extracellular data, and specifically for everything that's around spike sorting. So it won't load data which are like patch clamp, or it would work with the juxtacellular data, I guess.
Right.
But I don't think you would need a spike
just
sorter.
one electrode spike sorter.
Exactly. You don't need a spike sorter for juxtacellular recording, since signals should be quite clear.
All right. Okay. Next, so how do the different spike sorters treat, planar MEAs versus the shank electrodes? Would every spike sorter okay, that's the next question. So maybe the first one. How does it treat planar MEAs versus the shank?
Yeah, so basically the main difference is only in the here. It's only in the geometry, right?
Mm-hmm.
And the geometry, both for the planar and the shank MEAs, it's inferred from the file format. So the geometry is part of the MaxWell data files, and it's also part of the NeuroPixels data file. So in that case, you wouldn't need to upload the prop file. And then, of course, the spike sorters will make use of the position of each electrode to, for example, cluster the different spikes, to run this template matching step on the data. So these are used internally by the different spike sorters. I would say in general, high density helps, but also makes spike sorting a little bit more harder to be efficient, and it could be prone to find more units because you have a lot of the units appear of many different channels, right? So one error that could happen is that basically the main peak is here, but also you have smaller peaks here, and the template is not large enough or something like this, so you find some duplicate units. This could happen, but I would say that in general, we didn't see a lot of, let's say, differences also on simulated data if we use a high enough density, whether we have a planar or a shank configuration.
Right. Okay. And the next question, I think you already answered it earlier, but I'll just answer again. So the question is, would every spike sorter supported by SpikeInterface work on MaxWell's MEAs? And you already kind of answered it earlier.
Yeah, it should.
Support high density data sets.
It should, yes.
Yes. Thank you so much for this question. Next, we go to this one. Do you find that the false positives are the result of over-segmentation of individual spike clusters?
Okay, that's a good question.
From someone.
It is like someone that knows what we're talking about. Okay, so let me get back here. Marie, could you remove your video?
Yes. Goodbye.
Sorry. Yeah, so the thing is that we do compute this agreement matrix, right? So for each ground truth spike train, it might be that you have, like, instead of only one good match, you might have two good matches, right? So we are able to kind of classify a false positive unit, so meaning that a unit like this one that doesn't match well with any unit, so in this case, this would be below 5% or something. But if we have, like, imagine if this unit 8 would have like a large value for this unit 9, where the best match is 85%. Let's say here we had like 30%, then we are able to flag these units as being an oversplit, and we call this a redundant unit. On the other end, we can also classify the unit as overmerged, and this is the case for this unit 4. You see, you have one unit that matches with like around 30% accuracy agreement scores with 3 different ground truth units. So this is an overmerged unit because it merged the activity of 3 ground truth units. So by analyzing this agreement matrix, we're actually able to also like focus in detail of when spike sorters fail on which specific units, and also to try to improve the algorithms on exactly their failures, right? So yeah, but the false positive, again, for the question would be something like this, something that is not matched well to any unit.
All right. Thank you so much. I'm back again.
Okay, hello.
All right. Yes, we have one last question. The rest, thank you so much for all the questions. We will, if in case we're not able to answer them, we will send by email. Okay, so the last one we have is this one. Are there any deep learning based sorters?
Good question. So there have been some attempts to make like fully deep learning based spike sorters. The problem with deep learning in general, it's not a problem. Like the feature and the problem is in this case is that in order to train these deep learning models, you need like label data, right? So you could imagine to try to build a full end-to-end deep learning based spike sorter based on simulated data, right? Like we used here, these are simulated data sets. The problem is that these simulated data sets, while they do replicate some interesting features of real recordings, they cannot replicate some specific aspects of the real, what's the real world out there, right? So of course, it might be possible to do it, but you would need to rely on some simulations in order to build the model. And so then it's going to be a little bit hard to validate it on real data. Another possibility would be to use deep learning for specific aspects of the spike sorting pipeline, and especially spike sorter that we recently included in the SpikeInterface is YAS version 2. And that uses actually some deep learning internally to clean up the features and to improve the detection step. So in these specific steps, basically also some relatively simple simulations could have shown to improve the accuracy.
All right. Okay. Thanks a lot. So before we end, maybe what's next for SpikeInterface?
What's next is trying to improve it all the time. It's actually a lot of work to give it. No, it's true. And so we have we're trying also always to include new file formats to improve the compatibility, to improve the the supported technologies in terms of spike sorting. But we also have a wide range of pre-processing and post-processing tools that we are trying to integrate and maintain. So one thing that we haven't solved yet with SpikeInterface is how to properly install the spike sorters. This can be very tricky, right?
Like one-click installation that all the packages.
This is quite tricky, especially for like the GPU-based or these like Spike in Circles uses this MPI library, which is a parallelization library. It's a bit complicated to install. And currently, this is completely left to the end user, but there are ways to do it. And actually, this is done in Spike Forest. So in Spike Forest, we used something that's called Docker. So Docker is it's a container that basically encapsulates an environment with all the dependencies of different spike sorters. So our next step and what we're trying to aim for is to basically allow to run this SpikeInterface on like cloud services and completely remotely, so we can maintain the installation of the latest tools only in one place and provide basically the users with just an interface to run spike sorting, visualize the outputs, maybe also curate their data remotely. So we could also gather data and information about curation to make it more automatic. This would be like what, maybe from here in 5 years. Yeah, maybe less.
But sounds like a really good project, and the field is really growing, and it's still very, very important now that we have more and more people doing extracellular high density MEA recordings. So
yeah, I only hope one thing that so in some paper, in a few papers I saw, spike sorting was done in the normal way point that was in the methods, right? I hope you don't write something like this anymore. I hope I showed you that spike sorting is actually a complicated and unsolved problem. So at least try to say what you have done, what software you used, what version you used, and so on, and also try to share your spike sorted data. I think it would help your the community.
All right. And anyway, SpikeInterface allows this to be easier for everyone.
Yeah. We try. Easier.
Right. Okay. Alessio, is it okay if I share my screen?
Sure. Let me stop.
Thank you. All right. So I would like to thank everyone again for joining us today in this webinar. And thank you so much, Alessio. Thank you so much as well, Szilard. And yes, so just a few announcements on MaxWell Biosystems side. You can contact us at info@mxwbio.com in case you have questions, etc. You can schedule a call through Calendly and also visit our website. And most importantly, we are actually going to have a user meeting on April 22nd to 23rd, and we will send the invitations very soon. Please wait for that. We are very exciting speakers and also panel discussions upcoming. So thank you so much, everyone. Have a nice morning, evening, noon, wherever you are. Thank you so much. Bye.